AS Level Biology 9700, Paper 1, Variant 3, October, November 2024, question number 1 to question number 10. So, students, we will start from question number 1. So, question number 1 is, which steps are needed to find the actual width of a xylem vessel viewed in transfer section using a power objective 10 objective lens. So here you can see they have given us these 4 statements and we have to find out which statements are correct, and then according to the correct statements we will choose the correct option A, B, C or D. So first statement is convert from millimeter to micrometer by multiplying by 10 raised to power minus 3. And students, that when we convert the greater unit into a smaller unit, then we multiply it. So they have written multiply it by the 10 raised to power minus 3. When we convert millimeter into micrometer, then we multiply it with 1000 or 10 raised to power 3. So we will convert it with by multiplying it 1000 or 10 raised to power 3. So this statement is not correct because they have given 10 raised to power minus 3. So second statement is calibrate the eyepiece reticule using a stage micrometer on a power 4 objective lens. So power 4 objective lens we do not use for calibration. We use power lens 10s of the objective lens. So this statement is also not correct. The third statement, measure the width of the xylem vessel using an eyepiece reticule. Eyepiece reticule, this is the scale which is present in the eyepiece. So with the help of this eyepiece, we always measure the actual width of the xylem. So width of the xylem will be measured by the eyepiece reticule. So this is the correct statement. The fourth statement is multiply the number of eyepiece reticule units by the calibration of the eyepiece reticule. So multiply the number of eyepiece reticules which you have measured. You will multiply it by the calibration of the eyepiece reticule. This eyepiece reticule. So this is the correct statement. Fourth statement is correct statement. So it means last two statements 3 and 4 are correct. So according to these statements, we have to choose the correct option. A is not correct option because they have given the 4 statements into it. So this is not correct option. In B, they have given first 1 and 2, which are wrong. 3, 2, 3, 4 in 3 and 4 are correct, but 2 is not correct. So 3 and 4 are correct. So the correct option is D. Which cell structure is found in human cells and typical plant cells? So the which cell structure? 70S ribosomes. So these 70S ribosomes are present in human cells and in plant cells. So, according to this is the correct option. And secondly, they are present in eukaryotic cells, or you can say that cilia. They are present in human cells, not in the plant cells. And plasmodesmata are present in the plant cells. And tonoplast is the outer membrane of the sap vacuole. So tonoplast is only present in plant cells. So according to this, the option A is the correct option. Moving towards the question number 3, and question number 3 is, which statements are correct for a typical prokaryotic cell? Prokaryotic cell, here we can give an example in which the membrane bound organelles are not present. So they have given us 4 statements. It contains 70 S ribosomes. Yes, they contain the 70S ribosomes. They are present. Uh, it contains a cellulose cell wall. No cellulose cell wall. Instead of cellulose cell wall, they contain a cell wall of peptidoglycan. Peptidoglycan. So the cellulose cell wall is not present in prokaryotes. It contains circular DNA. Yes, plasmids are present in the form of circular DNA. It is up to 5 micrometer in diameter. Yes, this is the measurement of a maximum measurement of a prokaryotic cell. So according to this, the statement 1, 3, and 4, they are correct. Correct. So according to this, A is not correct, B is not correct, D is not correct. Only C is the correct option. Now question number 4. The diagram shows a typical animal cells and what is the function of the membrane system labeled X? They are endoplasmic reticulum. Rough endoplasmic reticulum because ribosomes are present on them. These dot-like structures are ribosomes. So, what is the function of the membrane system labeled X? Lipid synthesis on. This is the function of smooth endoplasmic reticulums, not the rough endoplasmic reticulum. Protein synthesis and transport. Yes, this is the function of rough endoplasmic reticulum. You know, the rough endoplasmic reticulum, ribosomes are attached on them. So function of them is the protein synthesis. And when the protein is synthesized, it is transported by the endoplasmic reticulum. So B can be the correct option and C is protein synthesis only. Beside protein synthesis, the transportation is also its function. So this is not the correct option. And D is transport only. No, it not only transport, but it also synthesize the protein. So only B is its correct option. Protein synthesis, ribosomes are over there. The protein synthesis takes place on rough endoplasmic reticulum, and after the formation, after the synthesis of protein, the protein is also transported. So the transportation of protein is also takes place by this rough endoplasmic reticulum. So B is the correct option. Question number 5. Question number 5 is what describes a lysosome? A vesicle containing enzymes enclosed by a double membrane. The vesicle containing enzymes, that this is correct. Enclosed a double membrane? No, they contain single membrane. That is part of the endoplasmic reticulum. Yes, and this is also incorrect because the lysosomes do not bud off from the endoplasmic reticulum, they bud off from the Golgi apparatus. So this statement is not correct. A vesicle containing hydrolytic enzymes, they contain hydrolytic enzymes and surrounded by a single membrane, found only in hydrolytic enzymes surrounded by a single membrane, found only in phagocytes. Phagocytes. This is not correct option. Phagocytes. Uh, only phagocytes. The hydrolytic enzymes, uh, they only phagocytes, so they are not only in phagocytes. They found on this is because phagocytes like macrophages and neutrophils are rich in lysosomes. due to their role in engulfing and breaking down foreign particles. So lysosomes are present almost in all animal cells, not only in phagocytes. So this is not the correct option. A vesicle enclosed by single membrane containing several different hydrolytic enzymes. They may act inside or outside the cell. They are secreted inside the cell. They for the breakdown, but they also uh, sometimes they are also transported outside the cell. They are released outside the cell in certain situations. So I would like to say this is the correct option. C is the correct option. A vesicle double membrane, again they have done written double membrane, so this is not the correct option. Only C is the correct option. That is a vesicle enclosed by single membrane. Single membrane is there because the double membranes are present in the nucleus, in the mitochondria, in the chloroplast, but not of this vesicle, which is called as the lysosome. So they contain single membrane. They contain single membrane and several different hydrolytic enzymes. So they are secreted inside the cell as well as outside the cell in certain situations. So according to this, the C is the correct option. Question number 6. Question number 6 is on screen now. The diagram shows a triglyceride. You can see the triglyceride over there. So they have given us, the fatty acids, 1 glycerol and 3 fatty acid molecules. This is saturated fatty acids. It do not have a double bond or triple bond inside it. So it is also a saturated fatty acid. Again, it do not have, so this is the unsaturated, and both of these are saturated fatty acids. Chains. So this is the 1 glycerol molecule. So according to this, we have to choose the correct option. What will be the product after hydrolysis of this triglyceride? After hydrolysis of this triglyceride, one molecule of glycogen, the totally wrong option. Glycogen is not a fatty acid. It is a polysaccharide which is formed from the glucose. So this is not the correct option. Again, the C option also, they have one molecule of glycogen. So this is not the correct. So B, they have one molecule of glycerol, 2 saturated fatty acids and 1 unsaturated fatty acid, as I have already written over here, because the triglyceride structure is actually triglyceride is an ester derived from one molecule of glycerol and 3 fatty acids. So the glycerol molecules has 3 hydroxyl groups. Each fatty acid has a carboxyl group, C double O, C double. So I would like to say in a triglyceride, the hydroxyl groups of glycerol are esterified with the carboxyl groups of the 3 fatty acids. So hydrolysis, when there will be hydrolysis, there will be the breakdown, and by the addition of water molecules. So here, this is the can be the correct. Now the D option is 1 molecule of glycerol, 1 saturated fatty acid, and 2 unsaturated fatty acids. 1 saturated, 1 unsaturated, and 2 saturated. So this is 1 and these are 2. and 3 molecules of water. Molecule with the addition of water, it is broken down. So water is added, not released. So D is not the correct option. Only B is the correct option. Next question 7. Question 7 is, which description lists all the components of a human hemoglobin molecule? 4 polypeptide chains. Yes, 4 polypeptide. 2 alpha and 2 beta chains. 2 alpha and 2 beta chains, and all chains contains one heme group. One heme group. So you can see the 4 polypeptides that are all same and one heme group. They are not same. They are different. So 2 alpha chains and 2 beta chains, they are different from each other. So 4 polypeptides that are not all the same and one heme group. One heme group. They contain 4 heme groups. For 4 polypeptides that are all the same and 4 heme groups, they are not same. Again, these are not the correct options. 4 polypeptides that are not all the same and 4 heme groups, this is the correct, that is they have 4 polypeptide chains, 2 alpha chains, 2 beta chains, and each chain contains its own heme group. Its own heme group to carry the oxygen molecule. So D is the correct option. Next, HIV-1 protease. HIV-1 protease is an enzyme produced by the HIV virus. So hemoglobin, two chains of 99 amino acids form the enzyme. In each chain, amino acids 25, 26, and 27 in the sequence form part of the active site. 99 amino acids will make enzyme, and enzyme amino acid number 25, 26, and 27. They will be the active sites. Active sites where the substrate attach itself on the enzyme. So this is for the attachment. So it means which order of protein structure control the shape of the active site. So there should be a proper sequence. And the hemoglobin is a globular protein which is found in the red blood cells, and it is responsible for the transportation of oxygen into the body and other parts, into the different parts of the body, you can say that. So it is a quaternary structure. Quaternary structure is made up of multiple polypeptide chains. So significantly, we can say that two we can say that in this case, you can say the 4 chains, but here in this case, you can see primary, secondary, tertiary, and quaternary structure. So I have quoted the example of this in this case. That is, in hemoglobin, there are 4 chains, but in the next question, the there is an enzyme. Enzyme is also a protein. Hemoglobin is a protein. Enzyme is also a protein. There are 4 chains, but here there are 2 identical chains. So it means here the heme group is present, but here in this case they have there are the active sites where the substrate will attach. So in this case, I would like to discuss the different shapes of these proteins. First of all, the primaries, I have discussed the quaternary structure, four polypeptide chains in tertiary, the three chains or and these two tertiary and quaternary will three-dimensional shapes. Three-dimensional shapes are proper shapes, and primary is a linear, you can say chain. Primary chain is a linear chain, and secondary chain consists of the alpha and beta plated sheets. So different types of these four different types of proteins gives the different shapes to the proteins. So alpha helicals or beta pleated sheets in the secondary, primary chain, and in this way tertiary, the three-dimensional shape. And you can see the quaternary stage proteins, more than one polypeptide chains or four polypeptides, in which I have also given the question number 7's example. So it means when there is an enzyme and there are two identical chains, in that case, all these four different types of proteins are involved for its maintenance. So A can be the correct option, primary, secondary, and tertiary only. No, it will not give the complete structure. So primary structure, specific amino acid sequence, so directly determines which R group. When there is primary only structure, so it will determines the where the different types of structures is there. So both four types of uh, proteins are arranged in a sequence. It will give the structure to an enzyme which is HIV1 protease. So according to this, four types are involved: primary, secondary, tertiary, and quaternary. This is the order of the protein structure which will control the shape of the active site. So we are moving towards the question number 9. Which statements are correct? Reasons for how animals cool down in the hot environment using latent heat of vaporization. So first of all, what is latent heat of vaporization? So latent heat of vaporization of water, it refers to the large amount of heat absorbed when water changes from a liquid to a gas. So when the water will change from liquid to a gas, so when water evaporates from a surface, so it takes this heat energy with it, leading to a cooling effect on that surface. So this is a primary mechanism for cooling in many animals. So here they have given us four statements. Some animals lie down and roll in wet soil. So this is the correct statement, because when animals roll down uh, into the wet soil, the water from the soil gets onto its body surface. So as this water evaporates from the animal's skin, also it absorbs latent heat from the animal's body. So thus cooling the animals, it will give the cooling down or the cooling effect to the animals. So this is the correct statement. Fish moves into deeper water. Fish will not move into the deeper water to cool down its body. So it will go in the moist, it will hide itself into the moist soil, not into the deeper water. So this is not correct statement. Some animals lick their fur to make it wet. Yes, this is just like the rolling down of the animals into the wet soil. So they will lick their fur with their you can say, and then when they will lick in this way, this statement is correct, because when they are licking to their body, so when this their saliva evaporates from the fur, so it will also absorb the latent heat from the animal's body, so it will also giving them a cooling effect. So this is also correct. So some animals breathe quickly with a wet tongue hanging out of their mouth, just like it takes place in the dogs. Uh, this is also a cooling mechanism in many mammals. When an animal's pants, air passes rapidly over the moist surface of the tongue and respiratory tract. So the evaporation of water from these wet surfaces will give them a cooling effect. So this is also the correct option. So according to 1, 3, and 4 are the correct options. So this is, according to this, C is the correct option. Next, question number 10. And question number 10 is two different enzymes P and Q. So this P and Q, two different enzymes, are investigated to find out the optimum pH for each enzyme. The results show that P works only in the acidic conditions. P is mentioned with a straight line. Only in acidic conditions. So it works in P acidic conditions. And Q with the dotted line, which works only in slightly alkaline. Which work in slightly alkaline. So we will move towards the graph 1. They have given on x-axis, they have given pH, which is an independent value, and rate of reaction will be changed when the pH will increase or decrease. So P is according to the statement, the P is working in the acidic condition and Q is in the alkaline. Whereas in this graph, you can see below 7, this is the acidic, and 7 to 14, let me in this way. 7 is a neutral value. So 0 to 7 is acidic, and acidity is increasing from 7 to 0. On the other hand, 7 to 14, the values are alkaline, and alkalinity increases from 7 to 14. So in this way, this is not the correct, because the P is only working in, and here the P is on neutral value and Q is in acidic condition. So A is not correct option. On B, you can see the substrate concentration at 1 minute. On x-axis, pH is there. On y-axis, they have given substrate concentration at 1 minute. And in 1 minute, how much of the substrate concentration is decreasing. So here you can see the maximum concentrate process is taking place in acidic conditions. And then as acidity is decreasing, the rate of substrate concentration is increasing. So on the other hand, at Q, you can also see slightly alkaline, more than 7 value, more than 7, between 8 or 14. So the value is between 8 and 14, so slightly alkaline. So substrate concentration is decreasing in slightly alkaline conditions. So it is the correct graph. It shows the correct answer. On C, you can see the pH is an independent value which should be on x-axis and it should not be on y-axis. So according to this graph is not correct. Again, the option D is not correct because they have taken the pH on y-axis. So according to that, they have given us pH. This is an independent value. So according to this, B is the correct option. So A, C, and D are not correct graphs. So it was students, it was all about question number 1 to 10. In the next video, I will discuss from question number 11 to 20. Please like, share, and subscribe my channel to watch more videos. And in some days, I will also upload my lessons of AS level, inshallah. Thanks for watching.